Well, welcome everybody. Welcome to the first webinar of BAMMsat on BEXUS. And we are some students who came together from different universities and different countries around the world to make some cool science and engineering. And we hope that we teach you and you'll learn something today about this very interesting project. So today we have Akil and Mike, who are going to be the main speakers today. We also have Dr. Cullen, who is the supervisor of the team, and Tomaso and me, Jesus, we are the outreach team inside BAMMsat. There are also multiple members who are present in the webinar in case you have like more technical questions, but let's begin. Akil, you have the floor.
Okay, well, first of all, I'm gonna introduce myself, but I'm gonna let Mike introduce himself. So I'm currently doing my PhD on BAMMsat. So BAMMsat will stand for something I will go into detail later on. So my PhD is really trying to push the current state of BAMMsat into actual real-life application in preparation for future space flight. Mike, do you want to just give a quick introduction?
Yeah, hi, I'm Mike Cook. I'm also a PhD. I'm down in Exeter, and mine's on healthy aging, which uses the model organism C. elegans.
Yeah. So I think we got together because we proposed a mission on the ISS at the first year of my PhD, and that's how we, Mike and I were introduced basically. And now at the end, in my third year, we came out with this project for the REXUS BEXUS flight. So introduction to the project, the experiment is called BAMMsat on BEXUS. Our team is called Bob. We are a 9 student from two main universities. We also have the master from the International Space Universities. And you can see we are a very multinational team. What we're trying to do is essentially trying to run an experiment on the REXUS BEXUS campaign program. So REXUS BEXUS is a European-wide program. The idea is that they allow university students to launch experiment on the sounding rocket or a balloon. So we are on the balloon side of things. And what we're trying to do is building essentially a small lab to perform biological type experiment in preparation for future space mission. So if I go on the next slide. So this is our team, just to put pictures of names. We have, again, multinational team, and we try to cover the different aspects of trying to build a hardware. So you have the power, of course, you have the system, the actual experiment payload, and yeah, biology or a payload, because you can't really build something without understanding the biology that you want to build in. And of course, the mechanical software. We also have a management. Is quite critical, especially at this time of the pandemic, but also REXUS BEXUS in general, from proposal to flight. So we're actually going to be flying this. Is a 12 months project turnaround. So it's a very, very tight space mission, basically. And of course, we want to try to put the stories out there, trying to share our project, trying to share our mission, and that's why we have community and outreach managers. Just a quick shout out to all the sponsors. So the REXUS BEXUS is a program basically run by 3 different organizations. The main one is the Swedish National Space Agency, German Aerospace Center, and also the European Space Agency. And we have a lot of support from different organizations in terms of grants and also hardware for us to run the experiments. So BAMMsat stands for something, and it stands for Bioscience, Astrobiology, Medicine Material on a CubeSat. So a CubeSat is a standardized satellite. It's basically a cubic satellite. You can visualize it as a shoebox. I mean, if you want to get a bit more representation, this is a CubeSat. This is a 3 unit, so 2 unit and 1 unit. So we try to basically build an experiment that can cover this field. We'll go into why we are doing this, but we're trying to basically build a flexible miniaturized lab. I think if you can remember one thing, just remember that today. So why are we doing this? So first of all, before I go into that detail, this is the ISS NanoRack deployer. This is a 3-unit CubeSat. Just to give you a bit more perspective how it will look like. So our whole design will basically be a combination of these 3. So the reason why we are doing this is space is very difficult. There's a lot of reasons why we want to go to space. It's for research, because space has microgravity in combination with high energy radiation particle, in combination with vacuum. So it's not really feasible to run all of that on Earth, and it's not really practical either. And the reason why we go to space is because this environment really enables us to push the limits of biology and fundamental science. So for example, we know that we can build better drugs in microgravity because the crystal just forms basically a lot better than Earth. There's no gravity pulling it.
One question. What's in the image here? What is this ESA Columbus thing?
Yeah, sorry, yeah. So the ESA Columbus is an ISS module. So it's a European Space Agency module to basically for astronauts to run experiment in space. And this is very huge. I think ISS is the size of sometimes something football, and it is big. Our experiment is small. It's a cube. So yeah. But what I'm trying to say is this environment really helps us if we really want to go back to Moon and Mars, because we need to understand the impact on the astronauts and also the associated biology. So all the food, all the microbiomes, all the bacteria that we carry with us. But most importantly, doing this experiment really benefits people on Earth. And this is because we know we can basically study these limits and really benefits in terms of medical properties on Earth. And I think Mike will go into a bit more detail to give you a biological perspective in a bit. So what do we need to be able to perform this, to be able to benefit from this? We need something that's cheap. So CubeSat is much smaller. It's not an ISS, so it doesn't take a lot of rockets to launch it, and it's lighter. So in space, mass is equal money. Again, another thing you need to remember, in space, mass is equal money. It needs to be for it to be repeatable. So something that's small and compact is something that we can be repeatable, and we need to have a high number of subjects, because biological experiment is not a small sample size type experiment. You can't populate 7.7 billion people on Earth based on one studies. Okay, so I think I'm gonna pass it to Mike now. He will give a biological side of things, and there you go, Mike.
So basically, space is very dangerous, and we keep hearing about these proposed missions to the Moon and to the Mars, and particularly to Mars, because we'll be in space for so long, it will basically equate to a suicide mission, because not only does microgravity cause like really advanced aging, about 1 year in spaceflight is equal to 40 years of aging on Earth, there's also a lot of radiation damage, and so there's high cancer risks. And we only have a very small amount of knowledge just by being in space for as long as we have been. And so we need to find really effective ways to intervene some of this damage, both on physical level and biological. However, this is obviously very expensive, and there's very little access to the scientific community in order to find these interventions we so desperately need. And we also need this to be high throughput so that we can make the most out of being in space, or else it will not be cost effective. There are ways that we try to simulate microgravity. However, these are not true representations, and you also don't experience the same radiation levels that you would. And so they're not ideal when we're trying to find actual interventions to work in a real life flight. We know about some of the more obvious changes in space flights. As you can see here, we have Mark and Scott Kelly, who did the twin experiment. And as you can see, they're identical twins. However, Scott Kelly on the left here, he has aged much quicker than his brother, who did not go into space.
One thing, Mike, I think Scott Kelly is the one in the right.
Is it?
Yeah.
Yeah.
Don't know my left lights. So these experiments are obviously very flawed, and we need to know a lot more before we do these deep spaceflight missions, particularly those to Mars. And also these human experiments have a very narrow scope. For example, when we think about this twin experiment, it's very expensive because you have to pay for the food, the water, the wages, maintaining the space station, all this all adds up. There's also very few participants available, and we have to wonder whether or not these actually replicate and represent the human population here on Earth, especially as astronauts are a very specialized population, having undergone a lot of training needed so that they are very physically and mentally fit, and thus very unlike the average population. And we have to also consider that there are very few astronauts at any one time, so even if we do these experiments, it's very hard to get reproducibility. Next slide, please. So if we want to truly understand the effects of spaceflight, we need to find alternative ways to find what's happening both to the whole organism and down to the cellular and molecular level, which is reliable, reproducible, and high throughput. For this, we believe that using alternative biological models or model organisms, and this will allow us to use much higher populations and high throughput intervention techniques, but it's sorry. But it's also necessary for them to have the same adaptations to spaceflight. C. elegans are a nematode worm which have previously been used in spaceflight for this very reason. They're very well suited due to their small microscopic size, only growing to about 1 millimeter. They also have a very short life cycle, meaning that they grow very quickly and they don't live very long, so that we can see an entire lifespan in the space of a few weeks. They're also very small and require very little maintenance, and as Akil's already said, in space, time is money and so is volume, so that all adds up to make it a much more cost-effective model. Next slide, please. They are also considered a suitable organism for studying human conditions, not just in space, but also on Earth. And this is because they have a highly conserved genome with us, which means that the building blocks of their cells are very similar to ours. And they also have highly conserved organ architecture. For example, their muscles are very similar to us. And this all comes from having very similar molecular and signaling pathways, and so they adapt to space in the same way that we do. On Earth, we use them regularly to study things like radiation biology and developmental and aging biology, as well as muscle adaptations, and they can even be genetically altered to respond to different environments and model human diseases. C. elegans have already given us a great insight into genetic changes that occur during spaceflight, and they provided potential mechanisms for why and how astronauts age so fast and lose muscle in a much faster rate during spaceflight. And this is leading to testing pharmaceutical interventions which may limit or delay this. They've also given us a lot of unexpected information, such as with neurodegenerative diseases such as Alzheimer's and Huntington's disease, where we've seen that these diseases progress much slower in microgravity. And all of this is very important because not only does it have impacts on the healthy aging and the health of astronauts, it also affects the aging population on Earth, as any interventions that we find that help astronauts will also help us as we age, as we will all experience the same and similar conditions. And back to Akil.
Yeah, so I think, I mean, in summary, I think C. elegans can help us age better, right?
One question, what is, I see some arrows in the images, what are they pointing at?
They're pointing at worms. So it's just to kind of give you a bit of scale, like they're very small worms. I think they've actually turned dour, so they've entered a elongating life stage process.
Yeah, so this is a healthy worm, this is a dour worm.
Yeah.
I mean, okay. A bit of fun fact, C. elegans is basically a normal nematode worm that you can find on the soil. It was actually first discovered at Bristol, am I correct?
Yep, that's right.
Bristol.
Yeah, and they're found all over the world.
And they're a very tough animal.
Very.
Okay.
I'm gonna go back into the hardware side of the thing. I'm not gonna go into much detail on the technology. Probably that will be in the next webinar. This webinar is just an introduction. But what we're trying to do while you're reading this text is essentially we're trying to perform technology and operation demonstration of the hardware. So we want to basically verify that it can work in a space-like environment. We want to develop all the pre-flight operations, because when you're handling biology, it has a lot more requirements than just doing a chemical study, for example. You need to make sure it's alive, it's fed well and such. And obviously, we want to be able to deliver an actual hardware at the end. this is to give you another flavor of the current stage. So we just passed our CDR as critical design review. We are coming up into our next review, which is in integration progress review. So this is at this stage. This is essentially what the hardware will look like. We have the microfluidics because we will be putting some administrating some drugs and preservative to the worms. The worms will be staying in this disk. We have a camera system to be able to record data on the worms. We have all the sensors for the fluidic side to make sure it's working properly. And all of this will be in a pressure vessel. And this is why we have a vacuum feed-through to connect to the 1U. So just imagine, this is a 2U, this is the experiment, and we need, it's in a vacuum, it's a pressure vessel, and connect that together.
How are they going to be connected? What's going to be the final configuration now that you have both structures there? Can you?
There will be mechanically mounted, so there will be bolts coming in from the 1U, and the electrical feed-through will be connected using this 50 pins vacuum. So power and communication data will come in through these pins.
Okay, very well.
Yeah, again, this is to give you a bit more flavor, basically what I just explained, and this is what we'll be launching in the large stratospheric balloon at Esrange in north of Sweden. It's gonna be in October this year, and it's gonna be very cold, so you can see ice everywhere. So hopefully, yeah, it will be fun. It will be great fun. It's a 10 days launch campaign. Just to give you a bit of flavor on the actual program itself. So as I mentioned, this is an opportunity provided by REXUS and BEXUS. It's been running on for 10, 15 years, I think. And the idea is that they want to train capacity for future engineers to actually fit in the space industry. And what we do is we will have a review at various points. So I think the fun one was at the Esrange Space Center. So we did a PDR, preliminary design. So we spent a week there. We have to defend our design. We have basically panels from European Space Agency, Swedish Space Agency, the German Space Agency. Basically a tough crowd. And you sit there trying to defend everything. But it's also fun. You learn a lot on the space program, how to run it, how to do it. And also you get to see the aurora. So this is the picture that we took at Sweden.
What are the two objects in the right hand corner?
The two objects on the right.
Yeah, there is like a big metal structure and also
yes.
a red worm.
Good point. So the worm is our mascot. If you follow our Instagram and Twitter and social media, basically, we will be using this to communicate the stories. This is the gondola. So remember the balloon? We were touching the experiment here. This is basically what we'll be carrying with other experiments. So for this there's a lot of box because one of the things that we needed to do during the PDR, the training week, is to negotiate with different experiments, basically where to put them, how does that impact other type of experiment, which is important in actual real life mission as well, because you need to negotiate where do you want to be launched in orbit, for example, that will affect the position where you're going to be placed in the rockets. So it's kind of giving you basically a flavor, and it makes a lot of difference when you actually get to see the gondola itself. You get to design it to be able to mechanically mount it on these things.
And
so, yeah.
What's the final position of the CubeSat, of our CubeSat?
Yes.
Where is it going to be?
Oh, where is it going? So it will be here. The reason why we put it here, there's two reasons. One, if this gondola during landing, it will be landing in some this kind of environment, so it's going to be quite hard landing. If you put it inside, it can be protected. And secondly as well, we put it on the corner because you can see it's away from other experiment, and we want to have various temperature loading, so variation like cold and hot and cold, to really simulate the in-orbit environment as much as we can, obviously.
So after you said that, all the teams wanted to put the experiments up so they didn't crash during the landing.
Yeah, I think, to be honest, they were quite happy with us. You can see our box is very small. All other experiments huge. So we actually have the least problem of everyone. So it was quite good. Okay. Yeah, this is our launch campaign. We will be keeping updated with this webinar a bit more often. So we are going to be telling our story as we go along. But during the launch campaign, we will have a full trying to show our experience as much as possible. So do follow us during that time. And thank you for your attention. We are open for question really. And again, this is very informal. That's the idea of a tea talk. So if you want to talk, you ask questions.
There is already a question from Raj Panchal from India and saying, what is the material of the structure of the CubeSat, the one that you have in the lab?
I will let our mechanical engineer to explain that, just to get in participating. Amin, do you want to join in?
Yeah, sure. Hi, everyone. My name is Amin. So basically, the material that we're using for overall structure is aluminium, the aluminium 7075 series. It has a tempered structure, so the strength is quite high. It's space graded. It's used in many CubeSats before. The structure of the P-pod deployment system as well as 7075 aluminium completely compatible with our structure. So hope that answers the question.
Mm-hmm. Yeah, if you have any other question, any of the attendees, you can write them in the chat. I wanted to ask you, Akil, what is the thing that you like the most, that excites you the most about this project?
To get a PhD. No, I mean, in all seriousness, I think, well, like I said before, it's challenging to go to space, and the reason is because the current facilities are expensive, it's big, takes a lot of resources, takes a lot of time. CubeSat really changing the demographic of space because it's standardized, it's small, it's cheap. I mean, it's cheap relative sense, but it's cheaper. So what excites me is that having this kind of platform where it's flexible, you know, we're trying to look at bioscience, astrobiology, medicine, material, we can really open up this platform for the science community, for everyone, start using it and really trying to push, you know, access to space, making it democratizing it, making it accessible.
Okay, we have some more questions. Maybe Mike can answer to this. How do nematodes differ to more traditional subjects like fruit flies?
It's kind of a difficult one, really, isn't it? Wow. Well, nematodes are a worm, so they have quite similar like organ architecture to us compared to a fruit fly. So like flies are obviously a lot more different to us. I think they're lower down in the hierarchy of organisms in relation to us. And so this gives us a much better representation. Worms are really good because we use them particularly well because of the link between mitochondria and muscle, which is very similar to what humans exhibit. And they also have added benefits to being clear, which a fruit fly, well, it can be, but is less clear. And so we can use things like GFP to express certain molecular targets in muscles and things, so we can visualize what's happening within a cell, which is much harder to do in a fly.
Another of the questions here, around what altitude will the project fly? Or in the stratosphere, right?
Yeah, so if you look at the gun, let's show some pictures. So obviously the maximum altitude will depend on how heavy that is. But we are expecting maybe 25 kilometers from ground. So basically just a bit higher than what planes are traveling. I think.
We have other questions like, will it be cold and snowy in Esrange in the lunchtime in October?
Yes, maybe no. I don't think so, to be honest.
You don't think so that in Sweden in October, that close to the polar circle, nothing?
I think climate change has an impact on that.
Okay.
So I asked the question just to kind of clarify, because you gave pictures showing Esrange in what looked like the middle of winter, when it definitely is cold, as you know, because you went out there. But in mid-October, it's at that transition between the seasons. And so it might be relatively mild, no snow, or of course it could be kind of the start of the snow season. So it's very variable as to at that time of the year whether it will be cold and snowy, or it will just be relatively mild and no snow. So both options are kind of possible at that time of the year.
Mm-hmm.
And this is where our challenge is because one of the key requirements in any biological type experiment is temperature control. Because if it's too cold, if it's too hot, they will die. So we have to accommodate for our particular launch. So for our launch from Sweden, it will be quite cold. So we can't let it go down to -10, which is what is expected at that time of the year.
Okay. Another of the questions is, what is BEXUS and what is REXUS? And I think you said at the beginning, but a little bit quickly, and it's a rocket or balloon experiment for university students.
Yes. So if the person that asking, if you're a student, you should apply for this. Yeah. So rocket is REXUS. B is for basically balloons.
Mm-hmm.
Yeah.
Are you launching from Esrange, and how long do you expect the balloon to last? Another question.
Well, it's the same as question as before. So there's few parameters that is in consideration. Basically, the balloon will be filled with a gas. So the gas needs to last as long as it can. But I think what worries the Swedish is that they don't want the payload to go to Russia. So it depends on the flight path during that time, the wind. So if it goes to Finland, it's okay. But if it goes to Russia, they have to terminate. So we are expecting on nominal, on average flight time is about 3 hours.
Mm-hmm.
Yeah.
One question: how would they terminate the flight? Do they have a control with the balloon, or they just like?
I think it's pyrocutters, right? Yeah, I think it's pyrocutters. Yeah. So they just put a hole in a balloon and it will go down slowly.
Mm-hmm.
Yeah,
it makes more sense. So
yeah.
Do you expect to test the drug delivery plus the reaction during BEXUS, or is there not enough flight time for that? That's a question for Mike.
Because it's just a technical demonstration, we've chosen to use sodium azide, which is an anesthetic. And if you've ever had surgery, you'll know like anesthetic works almost instantaneously. So we expect to see a reaction within 30 odd seconds. So it should be more than enough time.
Yep. Our current experiment timeline is about an hour. So we have about 2 hours. Oh, my battery is running out. We have about 2 hours a margin, basically. We should be able to perform a drug delivery experiment.
And how many teams were taking part of this? Of BEXUS, REXUS?
Oh,
like 10 or?
I think a lot. Like I think it's teams from all over Europe. I think there's 4 on our flight, and there's another 4 on. So basically, in October there will be 2 balloons, 4 on our platform, 4 on the other one.
And are all the topics about like bio, medicine, or astrobiology, or what do other teams do? Do you know?
Oh man, a lot. So if you go on the REXUS BEXUS website, you can see the different experiment that's on this year. But I mean, to just give you perspective, the first BEXUS flight, so balloon, was a winch mechanism experiment. But now it's actually very cutting edge experiment technology type of experiment. So it has evolved a lot, and you can see the value that is really put into the scientific community and also students.
Okay, another question: is BAMMsat project still have plans to launch their experiment into orbit? I don't think we are launching into orbit, right? Well, BAMMsat us, maybe.
Yes, I want to say yes. Maybe Dave can. Yeah, he said yes. So yeah, there's always plan to launch an experiment.
Yeah, so I think as was mentioned earlier, we're using the BEXUS opportunity to help in the development of the CubeSat payload with the anticipation of, yes, flying it in space in both LEO and beyond LEO. So this is very much a technology demonstration trying to kind of improve the TRL, the technology readiness level of the payload design by operating it in a more extreme environment than we would normally have in the laboratory, and also operating it in a space mission-like operational scenario with all the ground segments and the pre-flight and post-flight kind of activities included, such that we can then use the results of this to convince people in the future we are well prepared to then fight for a slot on some future launch opportunity, and also to get further funds to develop BAMMsat to the point where, yes, we can then fly it in LEO and especially beyond LEO.
Yeah. So this is where CubeSat is pretty good, because you can see it's basically a box. You can carry it on your suitcase. So if you have a big spacecraft mission to Mars, the Moon, we can just put it on there, then we can run experiment for that environment.
Mm-hmm. Another question here, I wonder who asked it. For a future deep space beyond LEO flight with nematode worms, what would the molecular pathways that would be targeted for a study to help understand the effect of deep space environment on humans, Mike?
I'm sorry, I should have just asked the question verbally.
Gosh. I mean, there's so much that we still don't understand. So I think it would be like half targeting things like the insulin pathway with antagonists, because we know that there's like a really mild form of diabetes develops. There's also things like how pains that we know are altered in spaceflight. And if we inhibit these, it's likely that the muscles are going to maintain their structure for longer. But I mean, we're still waiting on some of the results from MME, and that experiment has like a really in-depth gene sequencing aspect to it. And using that, that's going to give us a lot of pathways that we're going to want to interact with. And we should be getting those results, well, any day now. So once we have that, that will really help inform where we want to go with our next set of experiments. Yeah,
because the interesting thing is that I've always assumed, and somebody correct me if I'm wrong, I've always assumed that the microgravity environment in LEO and in deep space is effectively the same.
Yep.
So therefore, we're not going to be able to do any new science in deep space based primarily around microgravity, at least in isolation. The issue about going into deep space is we get beyond the kind of the relative protection of the Earth's geomagnetic field, and therefore it's a different radiation environment. And so I suppose if we were trying to build a science case to justify a beyond LEO flight with nematodes as representative of human kind of molecular pathways, we would want something that certainly had the more radiation sensitive pathways as a core aspect of the science case. It has always been my assumption.
Yeah.
Does that still seem sensible?
Yeah, like there are some like fantastic models in these worms where we can produce GFP, green fluorescent protein, and we can, based on the amount of radiation they're exposed to, affects how much basically they light up. And that's going to definitely be like a key aspect to seeing like how much the radiation actually affects the biology, because until we have much, until we really know how it's being affected and what it's doing, it's hard to know where to intervene.
Yeah, because obviously, for people who don't know, I've actually got a biosciences background. So my background is not actually engineering or space. My background is actually biosciences and bioscience technology. But you mentioned green fluorescent protein, or GFP. Obviously, for those who are not familiar with it, that's a fantastic tool, because what it does is it means that we can actually use that technique to target specific genes or kind of gene activities associated with all these molecular paths and molecular mechanisms, and such that if one of these genes is turned on or turned off, we produce a nematode worm that when the gene is turned on, it will perhaps produce, or it could produce, as long together with the product of the gene, it also produces a product which is this green fluorescent protein, which means that we can then simply see it with the optical microscope. And so we're actually looking at turning on and turning off particular genes by simply looking at the color change in the nematode worms. And so it's a very powerful technique to allow us to look at these mechanisms with actually a very simple readouts in terms of the engineering, a microscope. So I think originally the green fluorescent protein actually comes naturally from jellyfish, doesn't it?
Yeah, it does.
Yeah. So it's actually a gene from jellyfish that's used by the biosciences community routinely for these kind of things in the lab. But obviously we're trying to, or potentially going to exploit it in the future to allow us to do these kind of detailed studies, but in a very simple CubeSat payload in space.
Okay, let's continue with the questions. Thank you very much, Professor Cullen, for the explanation. Would you consider, here Joe asks, would you consider using more than one model or organism to rule out the possibility of any observed results being specific?
Yeah, we kind of plan on also using cell culture. So like this, this device will basically handle any biology that will fit within the disk, which means it can basically be used for, you know, worms, other nematodes, human cell culture. Like it's, you know, not very limited.
So as long as it fits in that hole there, we can put it. Our aim or our ultimate goal is really to put human in there. I mean, not the whole human, but human cells.
Mm-hmm. Do you have any specific launch vehicles in mind when the time comes for REXUS?
I can answer that. So we're not going to be launching on REXUS. We are going to be launching on BEXUS. But specific launch vehicle in mind, I think the REXUS are currently using the previous sounding rocket that the German has been having in stock for a while now. I think it's called the MAXUS rocket. If you go on their website, you get a bit more detail, but I think it's on the, it's called MAXUS, so MAXUS, I think. Yeah, it's basically the German have a lot of rocket in hand, so they just changed that into sounding rocket.
I'm gonna send here in the chat the link to the website. Yeah. So if anybody wants it.
I mean, who is Joe? I mean, you're a student,
no,
Joe?
That was Jamie. Jamie, he's from the astronautics course.
Student. All right.
And could you use BAMMsat to study human hibernation in space? From Professor Cullen, who has supervised a group design project in human hibernation too.
I think yes, right? As long as you can activate the molecular pathway for human hibernating. But the first question is, do we have the capability to do that? I don't think we do, right? I'm asking Mike and
the cosmos, the universe asking.
Really outside my field, I've got no idea.
Okay, so as kind of somebody said, kind of we've been looking at this in terms of the engineering consequences of potentially being able to make humans hibernate in space. Obviously, that's quite a common feature of science fiction kind of stories and movies. But this is being seriously now studied by both ESA and NASA, in that we know that other mammalian species, bears, marmots, all kinds of things, can go into hibernation. And therefore they can turn on, turn off genes that effectively massively reduce the metabolic activity of the mammals. And also, given that we know humans lose bone and lose muscle mass in space, in hibernating bears and such, they go completely inactive for many months, but all of the mechanisms which would normally cause the bones, because they're not being used and the muscles not being used, to be lost, all of those mechanisms are turned off, so there's no muscle or bone loss. So that's another reason why the space agencies are starting to look at this. And the view is that since most mammals have very similar genetics, the assumption is that the underlying molecular pathways and mechanisms that are present in bears must also be present in humans. It's just that through evolution, they've been turned off. And so there's now, in the space agencies, but also the medical community, people now looking to see if they can figure out how they could turn them on, well, what they are and whether they can turn them on in humans, so that in Earth applications, in medical kind of critical situations, you could put a patient into a hibernation state and therefore effectively stop the clock and have a much slower approach to solving a life-threatening situation. And of course, in space, obviously we could do what we do in science fiction movies at the moment. And so obviously BAMMsat could be used to start looking at some of these effects actually in space once the community has worked out what the targets are to look at. So that was obviously the basis behind the question.
Yep. Very well. Well, if there are no more questions, I think we have been like 45 minutes since we began. I think we have answered a lot of questions for like 25 minutes. So I think we should be quite proud of the stats of the webinar. And yeah, if you have any other question, we have a website, we have Instagram, LinkedIn, Facebook, we have everything. You can contact us and we will answer your questions. And also the webinar has been recorded and we will upload it. So if you had to leave at some point, you can rewatch it again.
Yep.
So thank you very much, everybody, and hope to see you in the following webinars.